In our previous slides, we learned about ATP molecules, biological oxidation reduction reactions, how electron motive force and proton motive force work in a biological system, and also about the flow of protons and electrons. We learned about glycolysis and how through a series of oxidation reduction reaction and isomerization reaction, we learned how pyruvate is generated inside the cytosol from a glucose molecule. Here is the net result of glycolysis. We saw that at the end of glycolysis, we were left with two ATP molecules, 2 NADH and two molecules of pyruvate. Now, if oxygen is available, the pyruvate can be broken down, or we can also say that it is oxidized all the way to carbon dioxide in cellular respiration, making many molecules of ATP. But if oxygen is not present and the condition is anaerobic, a pyruvate changes to a lactate by the process of fermentation. Now, since we are studying about oxidative phosphorylation, we will focus on the conversion of pyruvate into acetyl-CoA, which happens in the presence of oxygen or in aerobic conditions. So let's look at the breakdown of a pyruvate molecule to acetyl coenzyme A, as you can see in the equation and also on the figure on the right side. Now, in order for the pyruvate, the product of glycolysis, to enter the next pathway, it must undergo several changes to become acetyl coenzyme A, or simply acetyl CoA. This acetyl CoA is a molecule that is further converted to oxaloacetate, which then enters the TCA cycle, or the citric acid cycle, or we can also call it the Krebs cycle. Now, the conversion of pyruvate to acetyl CoA is a three-step process, and it takes place inside the mitochondria. The pyruvate enters the mitochondria with the help of some transport proteins, and once inside it, it converts to acetyl CoA by a enzyme dehydrogenase. Now, we can divide this conversion of pyruvate to acetyl coenzyme A into three steps. So, in the first step, a carboxylic group is removed from the pyruvate, releasing a molecule of carbon dioxide into the surrounding medium. Now we know that carbon dioxide is one carbon attached to two oxygen atoms, and it is one of the major end product of cellular respiration. The result of this step is a 2-carbon hydroxyethyl group, which is bound to the enzyme pyruvate dehydrogenase. The loss of carbon dioxide is first of the 6 carbon from the original glucose molecule that has been removed. Now this step proceeds twice for every molecule of glucose metabolized. Now you have to remember that there are two pyruvate molecules produced at the end of glycolysis. Thus, two of the 6 carbons will have been removed at the end of both of these steps. Now in step number 2, the hydroxyethyl group is oxidized to an acetyl group, and the electrons are picked up by the NAD molecule, NAD positive molecule, as you can see under the arrow. It results in the formation of a NADH molecule, which is actually the reduced form of NAD positive. The high energy electrons from the NADH will then be used later by the cell to generate ATP for the energy. Now in step number 3, the enzyme bound acetyl group is transferred to coenzyme A, producing a molecule of acetyl coenzyme A, or acetyl CoA. This molecule of acetyl CoA is then further converted to be used in the next pathway of metabolism, which is the citric acid cycle or the Krebs cycle. Now here is what acetyl CoA or coenzyme A looks like. Now you don't have to remember its chemical structure, but you need to remember few important things about acetyl CoA, since it's an important molecule. Well, acetyl CoA or acetyl coenzyme A is a molecule that participates in many biological reactions in protein, carbohydrate, and lipid metabolism. Well, its main function is to deliver the acetyl group to the citric acid or Krebs cycle to be oxidized for energy production. The coenzyme A consists of a beta-mercaptoethylamine group linked to the vitamin pantothenic acid through a amide linkage, and a 3' phosphorylated ADP. I hope you remember what is an amide bond. We talked about it in our series of lecture on amino acid and proteins. Now, just to revise, an amide bond is a peptide bond. It's a covalent bond linking two consecutive alpha amino acids from the carbon number one of one amino acid to the nitrogen number two of another amino acid along a peptide or a protein chain. Anyhow, coming back to the coenzyme A, the acetyl group indicated in blue color in the structure diagram on the left side, you can see of the acetyl coenzyme A. It is linked to the sulfhydryl substitute of beta-mercaptoethylamine group. This thioester linkage is a high energy bond, which is particularly very reactive. Now, hydrolysis of the thiester bond is a exergonic reaction. It generates about minus 31 kilojoules per mole of energy. Now, coenzyme A is acetylated to acetyl coenzyme A by the breakdown of carbohydrates through glycolysis and by the breakdown of fatty acids through beta oxidation. Acetyl coenzyme A then enters the citric acid cycle where the acetyl group is oxidized to carbon dioxide and water, and the energy is released and is captured by the ATP molecules and one GTP molecule of the per acetyl group. Now, we can say that citric acid cycle, where the acetyl group is oxidized to carbon dioxide and water, it releases energy and it is captured to form around 11 ATPs and one GTP molecule. Now, there are two scenarios. Uh, when we think about the things going on outside the mitochondria. Now, in the cytosol, at high glucose level, glycolysis takes place rapidly, thus increasing the amount of citrate produced from the TCA cycle. This citrate is then exported to other organelles outside the mitochondria to be broken down into acetyl-CoA and oxaloacetate by the enzyme ATP citrate lyase. We can also call it ACL. Now, this principal reaction is coupled with the hydrolysis of ATP molecule. While at low glucose level, coenzyme A is acetylated using acetate by acetyl-CoA synthase enzyme, that's also called ACS, and also coupled with ATP hydrolysis. Ethanol also serves as a carbon source for acetylation of coenzyme A utilizing the enzyme alcohol dehydrogenase. Degradation of branch chain ketogenic amino acids such as we have valine, leucine or isoleucine also occurs. Now, these amino acids are converted to alpha keto acid by transamination or eventually by isovarylyl coenzyme A through oxidative decarboxylation or by an alpha keto acid dehydrogenase complex. Isovalyryl coenzyme A undergoes dehydrogenation, carboxylation, and a hydration reaction to form another coenzyme derivative intermediate before it is cleaved into coenzyme A and a acetoacetate group. While inside mitochondria at high glucose level, acetyl coenzyme A is produced through glycolysis. Pyruvate undergoes oxidative decarboxylation in which it loses its carboxylic group as a carbon dioxide molecule, as I told you earlier, to form a acetyl coenzyme A to give a large amount of energy. Now, oxidative conversion of pyruvate into acetyl coenzyme A is referred to as the pyruvate dehydrogenase reaction. It is catalyzed by the pyruvate dehydrogenase complex enzyme. Other conversion between pyruvate and acetyl coenzyme A are also possible. For example, we have pyruvate formate lyase that disproportionates pyruvate into acetyl coenzyme A and formic acid. Now, at low glucose level, the production of acetyl coenzyme A is linked to beta oxidation of fatty acids. Fatty acids are first converted to acetyl coenzyme A, or we can now actually it's converted into acyl coenzyme A. This acyl coenzyme A is then degraded into four steps of through a step of four cycles of oxidation, hydration, oxidation, and thiolysis, which is catalyzed by four respective enzymes. Oh, we can name them as acyl coenzyme dehydrogenase, enolyl hydratase, and dehydrogenase and ethyolase. The cycle produces a new fatty acid chain with less number of carbons and acetyl coenzyme A as a byproduct. In this diagram, you can see that acetyl coenzyme A, when it is formed, it's doing a lot of stuff inside the cell. Now, inside mitochondria, pyruvate is converted to acetyl-CoA by the pyruvate dehydrogenase complex, or PDH, and the acetyl coenzyme A is further oxidized by the TCA cycle with citrate synthase, catalyzing the first step. In the cytosol, pyruvate can be converted to acetaldehyde by pyruvate decarboxylase, or we can also call it PDC. And the acetaldehyde can be further converted to either ethanol by alcohol dehydrogenase or to acetate by aldehyde dehydrogenase. The acetate can enter both the nucleus and the peroxisome, and in both compartments as well as in the cytosol, it can be converted to acetyl coenzyme A by acetyl coenzyme A synthase enzyme. In the nucleus, acetyl coenzyme A is used for histone acetylation, whereas in case of the peroxisome, it can enter the glycoxylate cycle through reaction with the citrate synthase. Acetyl coenzyme of the cytosol can also enter the glycoxylate cycle through the reaction with malate synthase. The net result of the glycoxylate cycle is the formation of 1 mole of malate from 2 moles of acetyl coenzyme A, and malate can be transported to the mitochondria for further oxidation. Now, this route is essential for growth on acetate or ethanol for species such as yeast. Now, cytosolic acetyl coenzyme A is used for the biosynthesis of lipids, either different fatty acids, by malonyl coenzyme A. Many valuable products can be derived from these two intermediates. The acetyl carbon of the acetyl coenzyme A are released as carbon dioxide in the citric acid cycle. Now, let's look at the conversion of acetyl coenzyme A to carbon dioxide. Acetyl coenzyme A links glycolysis and pyruvate oxidation with the citric acid cycle. In the presence of oxygen, acetyl coenzyme A delivers its acetyl group to a 4-carbon molecule oxaloacetate to form a citrate, 6-carbon molecule with 3 carbon groups. And during the first steps of citric acid cycle, coenzyme A, which contains a sulfhydryl group, as you have seen in the previous structure, the SH group, it is recycled and becomes available to attach itself to another acetyl group. The citrate then harvests and then remains to the extractable energy from what begins as a glucose molecule and continues through the citric acid cycle. We're going to discuss the citric acid cycle in the coming slides in detail. Now, in the citric acid cycle, the 2 carbons that were originally the acetyl group of the acetyl coenzyme A are released as carbon dioxide, which is one of the major products of cellular respiration, through a series of enzymatic reactions that we are going to study later on. For each acetyl coenzyme A that enters the citric acid cycle, 2 carbon dioxide molecules are released in the reaction that are coupled with the production of a NADH molecule from the reduction of NAD positive molecules. In addition to the citric acid cycle, which is named for the first intermediate formed, which is actually citric acid or a citrate, when acetate joins to the oxaloacetate, the cycle is also known by 2 other names. The TCA cycle is named for tricarboxylic acids because of the citric acid citrate, and isocitrate, the first 2 intermediates that are formed, are tricarboxylic acids. Now let's look at citric acid cycle in detail. Now the citric acid cycle, as I told you, is also known as Krebs cycle. That's because it was identified in the 1937 by 2 biologists. One was Hans Adolf Krebs and the other one was Arthur Johnson. While Krebs' contribution was more, that's why that's also called as Krebs cycle. Now, the Krebs cycle or the citric acid cycle in detail. Like the conversion of pyruvate to acetyl coenzyme A, The citric acid cycle takes place inside the matrix of mitochondria. Almost all the enzymes of the citric acid cycle are soluble, with the single exception of an enzyme that's called succinate dehydrogenase. It is embedded in the inner membrane of the mitochondria. Now, unlike glycolysis, the citric acid cycle is a closed loop, like you can see in this figure. The last part of the pathway regenerates the compound used in the first step. The cycle can be divided into 8 steps. The 8 steps of the cycle are a series of redox, dehydration, hydration, and decarboxylation reaction that produce two carbon dioxide molecule, one GTP, an ATP, and a reduced form of NADH and FADH2 molecule. This is considerable and considered as a aerobic pathway because the NADH and FADH produced must transfer their electrons to the next pathway in the system, which is going to use oxygen. Now, if this transfer does not occur, the oxidation steps of the citric acid cycle can also not occur. Now, note that the citric acid cycle produces very little amount of ATP directly and does not directly consume oxygen. Now, let me explain citric acid cycle to you in a stepwise manner. So, in the fourth step, you can see that it's a condensation reaction combining the two carbon acetyl group from the acetyl coenzyme A with the 4 carbon oxaloacetate molecule to form a 6 carbon molecule of citrate. Now, the coenzyme A is bound to a sulfhydryl group and it diffuses away to eventually combine with another acetyl group. This step is irreversible because it is highly exergonic and it requires a lot of energy to go in the back direction. The rate of this reaction is controlled by a negative feedback and the amount of ATP that's available. If ATP level increases, the rate of this reaction is going to decrease. And if ATP is in short supply, the rate of this reaction is going to increase. Now look at step number 2. Citrate loses one water molecule and gains another as the citrate is converted into its isomer isocitrate. Now step number 3 and 4. In these steps, for example, in step number 3, you see that isocitrate is being oxidized and it is producing a 5-carbon molecule and alpha ketoglutarate together with a molecule of carbon dioxide and 2 electrons, which reduce NADH positive to NADH. This step is also regulated by a negative feedback from ATP and a NADH and by a positive effect of ADP. Step 3 and step 4, these are both oxidation and decarboxylation steps, as you can see, and they release electrons that reduce NAD positive into NADH and release carbon dioxide or carboxylic group from the carbon dioxide molecule. Now the alpha ketoglutarate in this step number 3 and its succinyl group is the product of step number 4. Coenzyme A bind to the succinyl group to form a succinyl coenzyme A. The enzyme that catalyzes step number 4 is regulated by a feedback inhibition of the ATP, succinyl coenzyme A, and NADH. Now look at step number 5. You see a phosphate group is substituted for coenzyme A and a high energy bond is formed. Now, this energy is used in substrate level phosphorylation during the conversion of the succinyl group to succinate to form another GTP molecule, a guanine triphosphate, or an ATP molecule. Now, there are two forms of enzymes called isoenzyme for this step, depending upon the type of animal tissue in which they are found. One form is found in the tissue that use large amount of ATP, such as heart and skeletal muscles. This form produces ATP. Now, the second form of the enzyme that is on tissues that have a high number of anabolic pathways, such as liver, this form produces GTP. GTP is energetically, just remember that the GTP is energetically equivalent to an ATP molecule. However, its use is more restricted. In particular, protein synthesis primarily uses a GTP molecule rather than an ATP molecule. Now look at step number 6. Now in step number 6, it's a dehydration process that converts succinate into fumarate. Two hydrogen atoms are transferred to FAD, producing FADH2. The energy contained in the electrons of these two steps of these two atoms is insufficient to produce a NAD positive molecule, but it's enough to produce a FAD molecule. Like NADH, this carrier remains attached to the enzyme and transfers the electrons to the electron transport chain directly. This process is made possible by the localization of the enzyme catalyzing this step inside the inner membrane of the mitochondria. In step number 7, you see that water is added to the fumarate during step 7 and malate is being produced. The last step is in the citric acid cycle regenerates oxaloacetate by oxidizing malate. Another molecule of NADH is as a result produced. Now the products of citric acid cycle, you see that the two carbon atom come in the citric acid cycle from each acetyl group, representing 4 out of the 6 carbon of one glucose molecule. The two carbon dioxide molecules are released on each turn of the cycle. However, they do not necessarily contain the most recently added carbon atoms. The two acetyl carbon will eventually be released on later turns of the cycle. Thus, all 6 carbon atoms from the original glucose molecule are eventually incorporated into carbon dioxide. And each turn of the cycle forms around 3 NADH molecules and 1 FADH2 molecule. These carriers will connect with the last portion of aerobic respiration to produce ATP molecule. 1 GTP or ATP is also made in each cycle. Several of the intermediate compounds in the citric acid cycle can be used in synthesizing non-essential amino acid. Therefore, the cycle is, we can say that it's amphibolic, which means that it's both catabolic as well as anabolic.